Yeah, so it's a great pleasure to welcome Ullasa. He's at ISA Trivandrum and has been a faculty there for a few years now. And his lab, I think of Ullasa as a primarily somebody who works on butterfly ecology and evolution and has done really interesting work, particularly with understanding wing pattern development and evolution. But his lab has been, and people in his group have been working on many different, with many different organisms and exploring many different themes in evolutionary biology and ecology and behavior. And so I'm really looking forward to listening to Ullasa's talk today. I hope that many of you have also seen a poster on very interesting work with Mimulus from one of his students. So without further ado, welcome Ullasa and looking forward to your talk. And I think Ullasa has indicated that people are welcome to interrupt in the middle. So please just, just open your mic and ask questions as we go along.
Yeah, thanks, Deepa. Can I know what's the background of students? Very briefly, maybe Deepa, you could summarize.
I think there are many more biologists this year, but there are definitely people who come from a non-biology background as well. Okay. And I think the biggest demographic is PhD students of some stage or the other, but we have a large number of people from masters at the master's level as well.
Okay, so there are a lot of people who may not be familiar with the very basics of plasticity, for example.
Yeah.
What I've planned is to have the first half would be more of a teaching kind of lecture or interaction session, and the second part will be focused more on work that we've been doing in our lab. I don't need to finish all slides, and I would like it to be interactive, and Deepa will let me know when my time is up. But I think rather than waiting for questions at the end, I would like to, I'd be happy if there are questions in between, and if there are discussion points, you can raise them up as well. Right? Yeah. All right. How many of you have come across the term phenotypic plasticity? I think maybe I'll answer that. I think Deepa must have talked about it very briefly. I think she mentioned she would talk about it. So most of you would have come across this term. So before we get into the term, so what do you see on the screen now? Those who are familiar, those who have come across this example will know this. The others, can you take a guess?
Some larva.
Very good.
Some.
Yeah. On the right, can you see my cursor?
Yes.
You can. So on the right here, you see a caterpillar of a moth. This one here is also a caterpillar. And it may not be immediately obvious, even when you're looking at this magnified pictures. But imagine you're a bird. You feed on caterpillars. Caterpillars are part of your diet. And if you're flying past when you're foraging, it's quite easy for you to miss these caterpillars. That's because they're well camouflaged. In fact, what we're talking about is a special kind of camouflage. Camouflage is the same as Crypsis. This is called Masquerade. So in this type of camouflage, prey animals resemble objects that are not edible to their predators. For example, in this case, you have a lizard. You can see that it resembles dried leaves, and it's quite easy for a predator to mistake this for a dried leaf, which is an inedible object for that particular predator. Here you have a stick insect, which resembles leaves. So it's masquerading as a eucalyptus leaf in this particular case. You can see the antenna sticking out. Here is a caterpillar or a larva of a butterfly. So this resembles bird poop. So if it's sitting on a leaf and if you're passing by, you or a predator might mistake it for bird poop. Obviously, this works only for predators that feed on these caterpillars, not on animals which would feed on poop. Right? Now, coming back to these caterpillars, what is surprising about these caterpillars is that they belong to the same species. So here is the adult, and these are two morphs or two types of caterpillars which can both grow up from the same set of eggs. Right? So this is a temperate moth, and it has two broods each year. There's a spring morph, and this particular caterpillar feeds on oak plants. So during spring, in the spring brood, the caterpillars feed on oak flowers. So these are they have oaks have catkins. So there's a special catkin is a special kind of inflorescence, and those caterpillars that feed on catkins end up masquerading as catkins. Those that feed on leaves, and this happens in the summer, in the next generation. So these guys, they go on to become adults, they mate, lay eggs, and by that time, the catkins are not so abundant. And now they feed on leaves, and they end up masquerading as these twig-like caterpillars, right? They're masquerading as twigs. And it turns out that this is related to the tannin content in their diet. So you can give them artificial diet with varying concentrations of tannins, and you can get both the spring and summer morphs in the lab. You can take the same set of eggs, you can take a set of eggs laid by one female, randomly divide them into two groups. Each group is fed on a different diet, and you can end up getting these morphs. So these eggs, they're very similar in their genotype. Right? So the reason they have, they have these different phenotypes because of their diet. In other words, because of their environment. So this is a very good example of phenotypic plasticity. So phenotypic plasticity is the ability of a genotype to produce more than one phenotype depending on the environment. So when we talk about the environment, we usually talk about the developing environment, because when they face a particular environment, when they're exposed to an environment, they develop phenotypes a little later. So that's part of development. So sometimes it's also called developmental plasticity. So these terms are used interchangeably, although some authors do find a distinction between the two. And it's important to keep in mind that in biology, especially in ecology, the environment includes both the biotic and abiotic environments. So when we talk of phenotypic plasticity, it's not simply about the effect of climatic variables, but also other biotic variables. For example, we talk about diet. In this case, we're talking about an interaction between the moth and its host plant. So the host plant is part of the biotic environment. There are a lot of excellent examples of phenotypic plasticity if you do a search online. I'll take a few examples today. In this butterfly, there are two morphs. One is a wet season morph that you see on the left, and a dry season morph. So this is an African butterfly, Bicyclus anilana, but there are many other related butterflies in India as well. In fact, there are thousands of butterflies across the world which have this kind of wing pattern plasticity. So in these butterflies, typically in the wet season, there's a series of very conspicuous structures called eye spots. And in the dry season, the eye spots become very small, or they're completely missing. And there are other changes in the wing pattern, because of which they become very cryptic against a dry leaf background. So the idea is that, the theory is that in the dry season, there's a lot of dry leaf on the ground, and these butterflies are fruit feeders. So they feed on rotting fruit on the forest floor, and when they are feeding on fruits, they are cryptic against a dry leaf background. On the other hand, in the wet season, the background is predominantly green, and having this phenotype is not going to be effective. And the theory is that having these eye spots protects them against predation, because predators are distracted by these eye spots. So rather than going for the head and thorax, the main body, they're distracted by these wing margins. They're distracted by the eye spots at the wing margins, and they attack the eye spots instead. So the butterfly can escape with a torn wing, but the butterfly is still alive. So they have two very different strategies during the wet and dry season, and the wing patterns seem to match this strategy. And under laboratory conditions, you can grow them under different environments. For example, it's been shown that if they're grown at high temperature, they end up having well-defined eye spots, but when they're grown at cold temperatures, they end up having this morph. And it turns out that in the regions where Bicyclus anilana is found, in the wet season, temperatures are higher, and in the dry season, temperatures are lower. So that matches with, the experiments match with what's observed in the wild as well. If you talk about caste,
I have
sure.
a question.
Yes.
So in your previous example, I mean, if I ask from the perspective of a predator, won't a predator would have evolved to combat their adaptation to each of the condition, or to escape from the predation?
Okay. There's something called co-evolution, and you can look up this term, co-evolutionary arms race. So you're right. The prey animals are, they evolve strategies to avoid being eaten. Predators avoid strategies to be better at detecting and at consuming prey animals. And there could be selection pressure for the predators to be able to identify these morphs in their respective backgrounds. For example, it's possible that a predator could have a different search image in the wet season and in the dry season. Or in the wet season, a predator could learn that eye spots are not nutritious, because if it's pecking at an eye spot, it's only getting a piece of wing. There's no nutrition in it. So they may evolve to ignore eye spots and attack the body instead. So there's always a selection pressure on the predators, and it may have happened. But in this particular case, I would say it's rather unlikely, because it's not going to be one single predator. There will be multiple species of predators which are predating upon these butterflies. So it's rather unlikely, but theoretically, it's definitely possible.
Okay, thanks. Yeah.
So how is caste determined in social insects? Let's take bees, honeybees. Anyone? I'm sure you've heard of different castes in bees, workers, queens. So what's the difference between a worker and a queen in a honeybee colony?
queen is sexually active, worker should.
The worker does most of the work, goes and finds honey, pollen, takes care of the brood, and so on. The queen mainly reproduces, right? So genotypically, they are the same. The worker and the queen, they're both females, but depending on the diet. So you might have heard of royal jelly. So depending on what is fed when they're growing, they either turn into workers or queens. In fact, in many hymenopterans, so in Hymenoptera is a group that includes bees and wasps. So in many Hymenoptera, if a queen dies or if a queen is removed, some other individual becomes a queen. That is again an example of phenotypic plasticity. So if you look at caste determination in social insects, in a lot of cases, these are examples of phenotypic plasticity. Right? They are not examples of genetic differences or controlling caste. Right? They're not genetically determined, rather. Locusts are a big problem. Right? And they are a big problem when they are gregarious. So you hear of these clouds of locusts which take over entire agricultural fields, entire towns, in fact, and economically, they create a lot of damage. But locusts are not always gregarious. In fact, they're usually solitary, and when they're solitary, they look greenish. But when the density increases, they come in contact with each other physically more frequently. And the more the physical contact between these locusts, the greater is the probability that they turn into a gregarious swarm. Well, I'm simplifying this, but there's usually a threshold. So beyond the threshold of physical contact, they turn into a gregarious morph. And then they group together and they can migrate. In fact, under laboratory conditions, researchers have found that they can take a brush and brush parts of the locust's body when they're solitary. You can take this and just brush them, and this simulates physical contact, and this can induce swarming or gregarious behavior. So here it's the phenotype we're talking about here is gregariousness itself, but we also see changes in their body color. That also seems to change. Of course, it's also controlled by hormones. I think in this case, it's controlled by serotonin. So if they're injected serotonin, that can also induce gregariousness. All of us will be familiar with the concept of genes and alleles and Mendelian inheritance. So here, when you talk about genetic control, we're talking about the effect of genes or combinations of genes, interactions between genes, and so on. In phenotypic plasticity, we're talking not about these, but we're talking about the influence of the environment. So often, when you see a phenotype, you might be interested in asking whether it's genetically controlled or phenotypically plastic. We'll come to that shortly. In fact, even though you can discuss examples of plasticity, you can discuss a lot of interesting examples of plasticity. In fact, plasticity itself is extremely common. There are so many examples where plasticity itself is not interesting, so we don't discuss about it. But if you think about it, practically every phenotype is plastic. So any phenotype that is sensitive to the environment is phenotypically plastic. Right? So we talk about what determines a phenotype. There's the effect of the genotype, the environment, and there's a G by E, gene by environment interactions. So when we talk about the effect of the environment, we are essentially talking about phenotypic plasticity. And when we talk about genotype by environment interactions, again, we're talking about phenotypic plasticity. So most phenotypes are plastic because developmental processes are usually sensitive to environmental variables. For example, in plants, heterophyly. Heterophyly is variation in leaf shape. Heterophyly is extremely common. In this particular plant, when they're grown under terrestrial conditions, at higher temperatures, they develop this kind of leaf. At low temperatures, they develop this kind of leaf. The leaf shape has changed quite drastically. And this particular plant also grows underwater or when they're submerged underwater for pretty long durations. And underwater, it develops into a very different phenotype. You could take clones and grow them under these three conditions, and you'll end up getting these three morphs. So do you think this could be adaptive in nature? It could be. In fact, it's not known how heterophyly benefits plants. It's not really clear. I mean, you could think of some reasons why it's beneficial, but in general, heterophyly is not well understood. Rather, why heterophyly has evolved in plants is not well understood. Here's another example. Here, this plant, when grown under high light intensity, it grows into this kind of plant with a lot of leaves. And under low light conditions, it grows into this kind of plant. Again, you can take clones and grow them under two conditions. You end up getting these two phenotypes. In this case, you might say that there's a shortage of sunlight for photosynthesis, because of which the plants are smaller. So it's not necessarily adaptive. But if you look closely, the leaves are wider. And even though the whole plant is smaller, if you look at the leaf surface area in relation to the overall size of the plant, you will see that leaf surface area is more in this case compared to that in the first case. So this could be adaptive in nature because they might be enhancing their leaf surface area so that they can use more of the sunlight available or use more of the light available. I've not received any pop-up notifications for raised hands. In case there's a raised hand, please do speak up as well. Now, an important question, as I said, is to figure out whether a phenotype is plastic or it's completely determined by the genes, or in other words, whether it's completely genetically determined. So if you have clones, as you can have in the case of plants, what you can do is you can grow the clones under different environmental conditions and check whether the phenotypes differ or not. Or, for example, in the case of moth eggs, what you could do is you could randomly assign the eggs to different groups and grow these groups under different environmental conditions. You could do it with siblings, or you could just sample the population and you could take seeds from a population of trees. You could take all seeds, divide them into two groups, and grow these under different environmental conditions and check whether the phenotypes differ. Or in the wild, you see two populations. Let's say it's a plant, and you see a particular trait, let's say flower color, is very different in these two populations. And you want to know whether this is because of phenotypic plasticity. For example, soil conditions could be different, and because of this, they could have different colors, or flower color itself is completely genetically controlled, therefore, they're genetically divergent, because of which they have different flower colors. So in this case, you could do what is called as a common garden experiment. You could bring in seeds from the wild and grow these sets of seeds in a greenhouse, for example, where soil is the same, temperature and other factors are the same for both sets of seeds. And you can look at what's happening to the phenotype. If they end up losing the differences that you see in the wild, it tells you that the flower color variation you see in the wild is because of phenotypic plasticity. So these two kinds of experiments are commonly done to figure out whether phenotypes are plastic or not. Plasticity is, it's part of, and many phenotypes are plastic. You can talk about morphology, physiology, you can talk about behavior. For example, we talked about wing pattern plasticity in this butterfly, Bicyclus aninana. It turns out that the dry and wet season also differ in terms of their behavior. In the wet season, the females are choosing. So they choose males based on, so what you see on the upper panels are the underside wing patterns, and on the panel below are the wing patterns on the upper side. So it turns out that these butterflies, they choose their males based on the size of these central white dots. So in the wet season, the females are very selective. They don't like males who have very large white spots. They don't like males who have very small spots. So they like intermediate, the males which have intermediate spots. For those of you who are familiar with this is an example of stabilizing selection. In the dry season, surprisingly, the males become choosy. The females are not so choosy. The males are choosy. So the males choose females depending on the size of these spots. So behavior is a complex phenotype, but in this case, you can grow them under two different temperatures, and you not only get two different wing phenotypes, but you also get two different behavioral phenotypes. The behavior itself, it's not clear why it's useful for the butterfly, why it enhances fitness, why females should be choosy in the wet season and males should be choosy. It's not really clear. And there are two broad kinds of plasticity. One is irreversible. For example, if you look at the caterpillar morphs of the moth that we looked at, the masquerading caterpillars, once they take a developmental trajectory, if the developmental pathways to become catkin masquerade, if they're triggered, then it cannot be reversed. So they go on to develop into catkin mimics or catkin masquerades. And this is not reversible. But there are many other examples of phenotypes where the phenotype itself can be reversed depending on environmental conditions. For example, if you talk about acclimatization, when mountaineers climb mountains, so they take breaks. So they climb perhaps a few hundred or 1,000 meters, and they wait for a while for their bodies to get acclimatized. And the body does get acclimatized, and They do this, periodically, and when they come back, their bodies change again. So this would be an example of reversible plasticity, which is also sometimes called flexibility. And if you think about learning, we can learn, we can unlearn things, and this is essentially a set of chemical changes in our brain. And this would also be an example of reversible plasticity.
Ullasa, there's a question. Amir, do you have a question still?
Yeah, yes.
Yeah.
In the previous slide,
Yeah.
So what is the operational sex ratio like in the wet and dry season? Is there any correlation?
It's a very good question. As far as we know, in the wild, the sex ratio does not really change. I mean, that would be an explanation if you have more males or more females in different seasons. You might have this kind of, this could be adaptive, but there's nothing that's been observed in the wild. And all of this has been shown under laboratory conditions. And this particular species has been in the lab for a very long time, for hundreds of generations. So it's not really clear why that's beneficial. Yeah, that's a very good question. Of course, a phenotype can be both genetically determined and plastic as well. If you take skin color, so there are genetic differences across populations, across individuals. But then if I go out and stand in the sun, then my skin would become a little darker, right? And if I come back indoors and stay indoors for a while, for a few days, it can go back to its original color. So skin color in this case is both genetically determined and phenotypically plastic. So when is phenotypic plasticity adaptive? So as I said, a lot of phenotypes, in fact, most phenotypes are sensitive to the environment. It doesn't mean that any example of phenotypic plasticity is an example of adaptation, right? Phenotypic plasticity is adaptive when the environment is variable, and in this variable environment, in this heterogeneous environment, the phenotypes that develop in the respective environment, they have higher fitness compared to the phenotype that does not develop. For example, if phenotype A develops in environment A and B develops in environment B, then in environment A, phenotype A has higher fitness compared to phenotype B. It's only under that condition plasticity is adaptive. Let's take this example. Here we talk about the influence of the biotic environment. In this barnacle, it's an aquatic organism. So there are two morphs. So this is a aperture, this is an opening, and they are on the rock surface. So there's an attack resistant morph. We're talking about attacks from this predator, the sea snail, and this morph, where the aperture is perpendicular to the rock surface, this morph is attack resistant, and the other morph is not so resistant. And it turns out that when the predators are present in the waters, they develop into the attack resistant morph. When the predators are absent, they develop into the regular morph. And under laboratory conditions, you can release these sea snails in water, leave them for a while, and if you leave them for a while, then leave some chemicals behind. And you grow these barnacles in this water, and they sense these chemicals and they develop into the attack resistant morph. In fact, such adaptations are quite common. In water fleas, in Daphnia, different species have different protective structures. For example, in this species, they have these spines which can protect them from predation, and these spines are produced only when their predators are present in water. And in this case, they develop a much big, a helmet-like structure which can protect them from predation. The same thing that you see here. And in this case, they have the spiny tail-like structure as well as a spike on their heads, on their helmets. And there are several other examples where size itself differs. Now here's a question. So yeah, go on.
I had a question about this. So are all of these examples of irreversible plasticity, which you're talking about, the barnacles and in Daphnia?
Yeah. In this case, there would be examples of irreversible. Yeah. As far as
Okay, so if there are examples of irreversible plasticity, then do we know what is the kind of change in environment these see in the wild in terms of predation pressure? Like, does predation pressure, is it like some locations see a lot of predators regularly versus other locations hardly see any predators, or is there a change in predation pressure regular? And depending on either of them, how does that affect the adaptability of this?
Yeah, so it's a good question. So in the first scenario, you could have regions, let's say waters, where these predators are by and large absent or rare, and you could have other regions where the predators are more common. So in this case, this would be a perfectly good way to, this would be a very good example of adaptive phenotypic plasticity, because if the predators are there, they're always there. If they're not there, it's unlikely that they will come in when the fleas grow up. Okay, but you could have another scenario where the presence of predators keeps varying over time, over shorter time periods, during the course of, during the lifespan of a water flea. In such a case, this strategy, this irreversible plasticity, may not be so good. It may not be so optimal because they can have the wrong phenotype. For example, when they're developing, they develop a phenotype. If the predators are not absent, they may develop one phenotype, but if the predators come in, it may not be a good phenotype to have. Okay, so there are 2 points here. One is reversible plasticity is more difficult to evolve in terms of development. It's more complex because you need to have the mechanism to turn something on and turning something off. And if you're talking about structures like helmets and so on, you can grow these helmets, but getting rid of them may not be so easy. Okay, even if they wanted to, even if there's selection pressure to get rid of them, right? So, well, I shouldn't talk about selection pressure, but even if, let's say, it's better for this individual to get rid of this helmet, they may not be able to get rid of it easily. So that's one thing. One thing is that it's difficult to evolve. And okay, I think I'll stop there. Let's just talk about the difficulty in evolving, because I won't get into the other point. We will run short of time. Okay, so that to be a short answer for you, difficulty in evolving. Does that answer your question for now?
Yes, thank you.
We have another 20 minutes, so let me go to a few other topics. Yeah.
Yes, sir. Thank you.
Right. This is something for you to think about. If one morph is better against predation by the snail, so we're talking about this example, then why not produce that morph always? Why have phenotypic plasticity at all? Why not just produce an attack-resistant morph in all cases, irrespective of whether the snail is present or not? Then you would not need the genes which are sensitive to the environment. You don't need to have these complex developmental pathways and so on. You don't need to have these switches and so on. Yeah, you can think about that. And skip this part. So often when we study phenotypic plasticity, we look at reaction norms, where on one axis you have the environment, and on the other axis you have the phenotype. So if you take genotype C, across all environments it has the same phenotype, therefore this is not phenotypically plastic. Whereas phenotype A and phenotype B are plastic. You can look at the same for helmet height. So here, as predator chemical cues, they increase in concentration, helmet height can increase. So this would also be an example of a reaction norm. I think I'll skip this part. No, okay, maybe I'll just mention quickly. You have examples of temperature-dependent sex determination in many animals. In turtles, for example, depending on how deep the eggs are in the soil, they experience different temperatures, and depending on the temperature at which they're incubating, they can develop either into males or females. And in fact, the turtle mother can dig a deeper pit or more shallow pit, and it can control the sex ratio. So this is an example of temperature-dependent sex determination. This is also an example of phenotypic plasticity. In some fish, they can change their sex. If the sex ratio changes, for example, in a population of there are no males, some females can switch their sex. And again, the environment in this case is sex ratio, or the presence or absence of males, and the phenotype would be sex itself. Let me skip these parts and go switch towards the work we have done in our lab. So we have been studying pupal color plasticity in butterflies for quite some time. So in this butterfly, Mycalisus mineus, it's a common butterfly found in India. This is closely related to Bicyclus aninana, the butterfly which had eye spot plasticity. So as in the case of Bicyclus, eye spots in the species are also plastic. So depending on the temperature at which they grow, the eye spots are either present or absent. But what we have been studying is color variation in the pupae. So pupae are very vulnerable to predation. They can't move, and once pupation takes place, they are there. They are the mercy of predators. So you can imagine predation pressure is quite strong. Rather, selection pressure to avoid predation is very strong in terms of wing pattern, camouflage, and so on. On the other hand, butterflies, even if they're detected, they might be able to fly away. Right? But pupae are relatively more vulnerable. What we've found is that in this species, there's variation in pupil color. The pupae are either green or brown. And we wanted to check whether this is phenotypically plastic or whether it's genetically determined. You could have, for example, in the simplest case, you could have one gene, two alleles which code for either green or brown. Okay? You can imagine that, you can expect that the green pupae are better camouflaged against a green background, and brown pupae are better camouflaged against a brown background. This could be soil, or it could be dried up vegetation. So this was our working hypothesis. So we thought that we expected that these pupae would be phenotypically plastic, and depending on where they pupate, the substrate would influence or would determine their pupil color. So we've done some experiments, and we were able to show that pupil color is indeed phenotypically plastic. It's not genetically determined. And I'll talk about those experiments on phenotypic plasticity we've done. So one of our hypotheses was that brown pupae should be more common. All right, so sorry, this is a mistake. This should be more brown pupae at lower RH. So RH is relative humidity. So it tells you how much moisture there is in the air. So we thought when the air is drier, you have more of this brown vegetation. So brown pupae should be better camouflaged. So our hypothesis was that when we grow these butterflies at lower RH, we should get more brown pupae. And that was indeed the case. So here you have low RH, this is 60% RH relative humidity, and this is high RH. So when we grow them at low RH, the proportion of brown pupae increases, but not by much. It was a very weak effect. Then we asked whether pupil color is correlated with substrate. So as I explained, you expect green pupae to be better camouflaged against green leaf background, and brown against backgrounds such as soil and so on. And this is what we found. So here we have brown pupae here and green pupae here. These bars represent pupae and different colors, and the hatchings represent off-leaf pupation. You can see that brown pupae are almost exclusively off-leaf. Off-leaf means they're away from leaves. This could be the stem or it could be soil. In our experiment, we had other surfaces like plastic pots and nylon sleeves and so on. It turns out that brown pupae almost never are found on leaves. Green pupae are predominantly found on leaves, but some of them also go away from leaves.
Sir,
Yeah,
I had
sure.
a quick question over there. So, is there a reason that the number of brown and green pupae in this experiment are so different from each other? Is there a particular reason for that?
The number of brown, okay. In general, the proportion of brown pupae tends to be quite low. So in general, the proportion of, you get many more green pupae in general. So this would depend on the experimental conditions. Depends on what containers we provide, what temperature we rear them in, the relative humidity. There's so many factors, but usually in this particular species, brown pupae are more infrequent.
Sir, I had a question. This experiment is done in different seasons or different conditions, or in the same conditions, or
so these are common garden experiments in the sense that they're not really common garden, but they were done in controlled conditions. Same temperatures, same humidity and so on, this particular experiment.
For both of them?
Absolutely, yeah.
And so the thing that I was asking, that is there any biasness for any particular, for brown pupae? There might be like the condition might be more suitable for the brown pupae compared to the.
Not really. See, in the previous experiment, we manipulated one relative humidity. Apart from RH, everything else was the same. In this case, everything was the same. In fact, you grow them together on the same plant, and some of them pupate on the leaves, some larvae, some others pupate away from leaves, and they turn out to be brown.
Okay. Okay. Yeah.
Yeah. So we wanted to test whether density affects color. For example, there's this long-standing hypothesis that having melanin. So in this case, the brown color is because of the presence of the pigment melanin. So there's a hypothesis that melanin increases immunity. So we thought, what happens if you grow them at high density? Do you get more melanized pupae? In other words, do you get more brown pupae? So we first grew them in plastic containers. We had 2 treatments, 1 larva per plastic pot versus 5 larvae. And even though we had a very large number of individuals, we did not get even a single brown. So even at a high density, so anything beyond 5 larvae per plastic container, mortality was extremely high. So we could not go higher than that. So the highest that we could practically use in the experiment, even at that density, we did not have any brown at all. So this would suggest that density does not influence color. But we repeated this experiment in these sleeves. So basically these are, it's nylon mesh material, and you have plant inside, they grow inside these. And this is a growth chamber where light conditions, temperature, humidity are under some degree of control. And so we had 3 treatments. So you can focus on the left graph. So we had 3 treatments: low density with 7 larvae per sleeve, intermediate with 14 larvae, and at high density where we had 28 larvae per sleeve. So here we found an effect of density. Under high density, the proportion of brown pupae was considerably higher compared to what you see under low density. And this was the case in growth chambers, and we repeated this experiment in outdoor terraces where conditions are not so well controlled. Here again we found under high density they have more brown. It turns out that the brown pupae develop faster. Here on the y-axis you have larval developmental time, and this is brown pupae, and of course the colors indicate brown and green pupae. You can see that brown larvae have shorter developmental periods. So it's possible that in a given sleeve, the pupae that develop faster, they don't have space to pupate, or they want to pupate away from the leaves because the leaves may get eaten up by other caterpillars. So they go away from the leaves, and because they're going away from the leaves, they end up becoming brown. So that's possible, and it may not be anything to do with immunity at all, as we initially had expected. We've looked at pupal plasticity in related species. So these are related butterflies. These 2 species in the genus Melanitis, they have only green pupae. They have Iptima baldus, which has only brown pupae. And we have another species, Iptima hibneri, which has both green and brown pupae. So we did similar experiments as we did with Mycalesis milias. And here you again have pupil proportions. So here we have substrate choice. So in the 2 species, the 2 Melanitis species which have only green pupae, you see that pupation, off-leaf pupation is indicated by these hatchings. So off-leaf pupation is fairly rare. In the 2 species which have both green and brown pupae, you see that approximately half pupate away from leaves and half pupate on leaves. And this is 1 species which has only brown pupae. Off-leaf pupation is very high. Okay? The proportion of pupae on leaves is quite less. So there's this nice trend where pupation substrate choice is correlated with pupil color. Those species which have brown pupae tend to pupate away from leaves. Those that have green tend to pupate only on leaves. And those that have both tend to distribute across both leaves and off-leaf substrates. I'll ignore this part of the graph for now. See, we have another 5 minutes. Okay, so I'll skip this experiment and I'll come to the last part. So in another species, we have quantified pupil color. So in this species, we don't have just green and brown pupae, but we have an entire range of pupil color. So it's possible to take photographs and quantify color. So we've used an index called greenness index. So we can get this index using a simple formula after we get the red, green, and blue pixel values for photographs. So you can see that if you plot greenness index and frequency for pupae, and here you have low greenness index, so this would be very brown pupae, and here you have high greenness index, so these are very green pupae, and this is the entire spectrum. You see that there's a continuous distribution. So it's not really green or brown, it's more evenly distributed. So there's the same trend that it's a similar trend that we saw in Myceliaceous minius. Those pupae that pupate off-leaf, so here they're orange. So on the left you have data from wild conditions, on right we have laboratory conditions. So the off-leaf pupae have lower greenness index. So on the y-axis you have greenness index. So the off-leaf pupae have lower greenness index, which means that they're more brown. The ones on leaves tend to be more green. Not only that, green pupae, if you take only the green pupae and if you plot the pupil greenness index against the leaf greenness index, the leaf greenness index would be the greenness index of the leaf on which a particular pupa was found. Turns out there's a nice correlation. What it means is that green pupae, they can alter or they can develop into different grades of different shades of green depending on the color of the leaf itself. So if they pupate on a very pale green leaf, they can develop into a pale green pupa. Dark green leaf, they can become darker green. So they're able to sense these finer scale variations and develop a matching phenotype. I'll stop here. I'd like to thank two students, PhD students, Harshad Mayekar and Tarun Kishore Yunam, who did all the work that I presented today. I think I overshot my time. I may not have much time for questions, but I'll be happy to take questions over email or rejoin your session later on in the evening.
Yeah, we can continue for five more minutes.
Okay, sure.
So please ask questions. Okay, I have a question. The leaf greenness, that last plot that you showed, wouldn't that change as the leaf dries up? I mean, I don't know what leaves are sort of relevant and how long it takes for pupation.
Yeah.
Is that something to worry about?
That's a good question, but leaves do change. But the rate of change in leaf greenness is slow compared to how quickly they emerge. So they're there only for a few days, 3 or 4 days, and then they eclose. So by that time, we assume that leaf color does not change so much.
Okay, makes sense. Okay, I have one more question about the previous plot where you showed that the pupae that developed faster were more likely to be brown in one of your focal species. I thought it should be the other way around, that if the green stuff is gone, then the ones that develop later should be the ones sort of left leafless perhaps or something like that, like there's no substrate to pupate on. So I'm a bit puzzled by this trend.
Yeah, it's true, but I mean, you could theoretically expect the other way around as well. But then if a plant is completely defoliated, right? Then these larvae would switch to another plant. They would not be there. So on the other hand, let's say there are 10 larvae feeding on a plant. And out of these 10, let's say there's one that develops fastest. So this one would, because once it pupates, it can't move away. So it would want to choose a place where there's some protection. So if there are enough leaves, maybe it chooses leaves. But if there are very few leaves, there's a threat of being defoliated completely. So it moves away and pupates near the soil or the stem. So that's a working hypothesis.
So, I mean, it sounds like there are lots of potentially confounding or complicating factors, right?
Absolutely.
Like feed into it. So, which then brings you to my last question here. I'm wondering whether could this simply be a bed hedging strategy kind of situation where it's not matching, it's not adaptive for one particular microhabitat or situation, but it's more like let's have some brown pupae and let's have some green pupae, and as long as they're more or less in the right spot, they're better off. For example, if you do experiments where you only provide green substrate and nothing else, do you see a higher fraction become green compared to a situation where both are present?
Yeah, I mean, if you give them conditions where there's a lot of green foliage, right? So, for example, if you grow them at very low density, right? So relative to one caterpillar, there's a lot of green foliage. Then you tend to get almost completely green.
Well, I guess I'm a little bit wary of those concluding things from that, simply because they still have stems available to pupate, right? It's not that there isn't any stem around to pupate on or pots around. Anyway.
Yeah. Well, plasticity itself could be a bed hedging strategy from a bigger perspective. Right? So if they have only green pupae, then they're forced, irrespective of density, they're forced to pupate on leaves. Right? So now, once they have plasticity, they can expand their niche, so to say.
Fair enough. Okay. Anybody else have any questions? Pratyush?
Yeah, I had one question. I was wondering if the pupal color is determined only at the stage of moving from the larva to the pupa, or could it be in some way influenced by the history of the larva itself, like where the larva, the habitat of the larva, or what it feeds on specifically? So is something known about this?
So we have been doing some experiments. So it seems like the last larval stages are the ones that really matter most. The early larval stages don't matter so much. So that's specific to pupal color plasticity. And more generally speaking, when we talk about butterflies in general, or any insect that has a caterpillar stage and a pupal stage and so on. So the stage that is closest to, let's say if you're talking about adult plasticity, then the previous stage, that is the stage that is closest to the adult stage, that would be the best predictor of the adult stage. Okay? So for example, the period just before the adult stage would best predict the environment of the adult. Therefore, the period before the adult stage would be most sensitive to the environment.
Okay, thank you.
Okay. All right. Well, then we close the session. Thank you very much, Ullasa. That was an interesting talk. Thanks for all the information on plasticity as well. And I hope that people have a chance to look at your work and ask more questions later on. Thanks. Bye.
Thank you. Bye.